One of NHGRI's major research projects for many years has been the Encyclopedia of DNA Elements, or ENCODE projects. ENCODE, in its current funding phase, is a little past the midpoint. And our usual practice here at NHGRI is to bring you a progress report or update on all our major research programs. And Brent Gravely, who is a council member and a member of the ENCODE Consortium, will give that update.
Okay, thank you, Rudy. And thanks for everyone for joining, both in the room and online. And today what I'd like to do, I've been tasked with, I should say, is giving you an update on the state of the ENCODE project. And there are several reasons for doing this. One is just for general knowledge, but this really fits in with the ongoing strategic plan and the future decisions that NHGRI is going to have to make. So for instance, ENCODE is currently ending in, I believe, 21, and the strategic plan will not be published till the end of October 2020. And in order to, so decisions have to be made prior to the publication of the strategic plan about the future of ENCODE, whether to continue it, and if so, what that direction would look like. And if it is not going to be continued, then what areas of research should NHGRI decide to pursue? So that's some of the context for what I'll be talking about today. Okay, so just to put this into context, at the beginning of when the ENCODE project started, fairly shortly before then, the draft sequence for the human genome had been published and made public. And what we were able to do at that point, we were very good at identifying coding regions of the genes, but we were really bad at identifying regulatory codes for the genome. And even for coding genes, we were only really good at identifying those that had the similar properties to those of known coding genes. So if they had the same codon bias and the same evolutionary signatures, we could find them. And if they had other properties, it was difficult to even identify those. And I would say anyone would be very hard-pressed to simply, at that point, scan the genome and try and identify an enhancer or a binding site for a transcription factor. So that became sort of the impetus for developing the ENCODE project. This is a little challenging because I can't see my slides up here. So the importance of non-coding DNA are many. So first of all, genes are regulated through time in development and in space in many different tissues, health and disease, and in response to environmental exposures. So it's very important to understand how the non-coding DNA that exists within the genome is set up to direct this sort of regulation that occurs. And what's really important is many of these regulatory elements and even the genes themselves are only active in very specific cells and tissues. So for instance, you may be unable to identify even a gene if you don't survey the transcriptional activity of the genome in the correct cell type. Most of the common disease-associated genetic variants actually lie outside of the protein coding regions, which also makes sense since most of the genome is non-protein coding. And then functional information is really needed to interpret the role of genetic variation in human disease and to apply this to genetics in the clinic. So we have to understand what these functional elements are and what happens to them when their activity is disrupted. And I'd also like to point out as the honorary RNA person that RNA regulatory elements are also encoded in the genome, but they only act after they're transcribed. So the goal of the ENCODE project at the outset really has been to develop a catalog for all functional elements in the human genome and a select number of model organisms. So this is really going in and identifying all the enhancers and repressors, the transcription factor binding sites, all the coding genes, the transcribed regions, etc. We want to make all of this information freely available to the research community as a resource so that it can enhance everyone's research and enhance our understanding of the regulation of gene expression and the genetic basis of disease. So this has both basic science and clinical applications. And there are many different aspects of the project. So first, there's data generation, so making all of the data in a uniform manner, analyzing the data in a uniform manner, and then the last two are really what makes the impact of the project possible, which is the data repository so that we can make the data available to the community, and then outreach, which is getting the information out there to people so that they know that it exists. So this is the timeline for the ENCODE project. So there was the pilot phase, which was in 2003 to 2007. And this is back in the dark ages when there only existed microarray technology. And so in this part of the project, only 1% of the genome was interrogated for human. And that phase occurred, and so there was both the data production aspect and technology development. And then the second phase had added into this the human, but also the fly and worm mod ENCODE projects. And again, the grants for phase 2 were written before next generation sequencing technology was really broadly available. So it was really during this time, the phase 2 of the project, that things transitioned from chip-type experiments to DNA sequencing experiments. So the fly and the worm project went on along with the human, and then staggered a little bit, a mouse portion of the project was added on. And again, there was a technology development component to phase 2 of the project. Then phase 3 started in 2013 to 16. And then this project, again, had the data production groups and technology development. The mod ENCODE, so the fly and the worm projects were ended, although there was a continuing project called Modern, which is headed by Bob Watterson and Sue Selnicker and Kevin White, that is studying all the transcription factors in flies and worms, and that's been ongoing, sort of follow-up of mod ENCODE. But in phase 3, one of the things that was added in was this computational analysis group. So it was during the phase 2 project, became apparent that we needed more computational experts to come in and develop new types of algorithms to develop the analytical tools to study new things. And then the current phase, so phase 4 of ENCODE, started in 2017, and it will end in 2012. And again, this has the data production groups, computational analysis group. And the biggest change really in this one, which I think is a very important one, is these functional characterization groups. And so what these groups are doing are taking the analytical outputs of the data production groups and computational analysis. So for instance, the predictions of all the enhancers in the genome, and they're performing massively parallel functional assays to validate these types of elements. And so we can actually initiate a loop where you can predict what these elements are and then actually test if they are doing them. And then that will then inform additional analysis and data production. Okay, so this is a slide that just highlights the different types of assays that are ongoing in ENCODE. So starting on the left side, we have the assays that interrogate the three-dimensional structure of the chromatin. So these are the 5C, high C type assays going on. There's a variety of assays, so the DNA-seq, ATAC-seq, et cetera, that are going on to interrogate hypersensitive sites in the genome. There's a huge effort ongoing to perform ChIP-seq experiments, and these are used to query both the chromatin structure as well as the transcription factor binding sites throughout the genome. There's assays that are used to measure methylation status. Computational predictions are used to both impute data and to analyze data. And then both long and short read RNA sequencing experiments are going on to interrogate the full transcriptome of the genome. And then CLIP-seq type experiments are being performed to characterize the protein RNA interactions that occur. And so all of these assays then are analyzed in order to identify the regulatory elements that can act both as promoters and long range, which elements might contact which promoters, identify all the exons and introns transcripts and the binding sites of proteins for those transcripts. Okay, so this is a slide that depicts the data production up to the end of 2007. So this is through ENCODE 3. And so what you can see is there's been a massive amount of data that's been generated to date for these projects. So for instance, this purple box represents the transcription factor binding assays. This one here is RNA sequencing, so querying transcription. We have RNA binding proteins, histone modifications, et cetera. So at that point, there were nearly 8,000 assays that had been generated and made publicly available. And just to highlight a few, and I will blatantly showing our data because I know it the best. But this is some data that we generated for ENCODE 3, which was a new type of data in the ENCODE project at that point, was studying the RNA binding proteins. And so we've generated a large data set of about 1,200 experiments. We're querying 356 RNA binding proteins. And throughout the genome, we're characterizing where these proteins interact with the RNA. We're asking what they do by performing experiments where we knock down the protein, followed by RNA-seq. We actually look at where within the cell those proteins reside. And we're also performing in vitro binding experiments so we can characterize the relative affinity of these proteins for all possible sequences. And so just one highlight where this data can actually be used to make predictions of how genomic variants may impact human health. This is intersecting the ENCODE data with the exact variant database. So this is a gene UTRN1, and there's a variant from the exact database that disrupts this G to a C, and that converts this sequence from the best possible binding site for a protein called RBFOX2 to the worst possible binding site for it. And there's an actual clip peak, a very strong one, right over this element here. And if you look then at the knockdown data, we see that this exon is strongly included in the control cells, but when you knock this protein down, you see skipping of that exon. So you can make a prediction now that this exact variant might disrupt the binding of this RNA binding protein, which would change the splicing of that exon. So that's the kind of information we're hoping to be able to use this data for to move things forward. Okay, so one of the big things that's been was initiated during ENCODE 3 and is continuing in ENCODE 4 is converting all of this data that's been generated into what's called the encyclopedia. And so this is the consortium's attempt at taking all this data and distilling it down to all of the elements so that you can simply look up these different elements and you'll have some information about what those might do. So this is done in many ways. We have what we call the ground level analysis efforts. So these are the individual assay results. So for instance, the RNA binding motif binding sites, the same for transcription factors, all the chromatin marks, et cetera. And so we make lists of all the elements queried by those. And then we have these integrated outputs. And so this is using all of the, for instance, chromatin marks, the DNase hypersensitive marks, et cetera, to identify promoter-like sequences, enhancer-like sequences, CTCF binding sites, and then genes that are linked together. And this is then used to develop what we call the registry of candidate cis-regulatory elements. And this is output into a database called SCREEN, which I'll talk about a little bit later. And this can then be viewed on the Santa Cruz browser directly. And I also like to point out that all of this data in ENCODE is now processed through uniform pipelines. So for every single data type, you don't have every individual lab processing their data using their own software that's specialized. Rather, the data production groups actually upload their raw FASTQ files to the data center, which then performs, if it's an RNA-seq experiment, the same exact analysis on all RNA-seq experiments using the same versions of the genome and the software, et cetera. So you get the same exact output for every single experiment. And all of this data is also from the ENCODE website. You can click a single button and it will send it to the browser and you can visualize it there. Okay, so there's many participants in the current phases of ENCODE. So there's all the mapping center PIs, some of which were previously part of ENCODE 3 or earlier, and some newer ones that are developing assays such as the 3D interaction data. There's the functional characterization PIs, as I mentioned. I think this is an earlier version of the slide, which is somehow 2 of the PIs got cut off, so I apologize. So there's the functional characterization PIs. We have the computational analysis PIs here, and then the data analysis center, or the DAC, and then the data coordination center. So this is Mike Cherry's group, and he does most of the outward-facing stuff that people can go to and actually download the data. So in addition to those core members, there are affiliated members. So there's a process by which people outside of the consortium can become part of the consortium, and they can participate in all of the conference calls and rules that are part of the ENCODE consortium. But just being colleagues with the individuals within the ENCODE project. So my lab is now, along with Jean Yao, we have a U41 community resource project to continue studying the RNA binding proteins, and we've become affiliated members. So we are now, even though we're not necessarily funded by ENCODE, we are now part of as an affiliate, and we still put our data into the ENCODE portal, make it freely available, and we play by all the same rules that everybody in the ENCODE project does. So there's our group, and then these are 2 computational groups that are also part of it as affiliates. So if you're interested in becoming affiliates, you can contact Elise Feingold or Mike Pazin or Dan Gilchrist, and they will give you all the information about that. And so the organization of this is that we have the mapping centers, which do the bulk of the data generation. We now have the functional characterization centers. So these are the groups that are doing these massively parallel assays to test enhancer predictions, et cetera. All of their data goes into the data coordination and analysis center, and then the computational analysis groups work with them to analyze the data, and the processed output is then incorporated into the encyclopedia. Okay, so one of the, I think the biggest or most important roles of the consortium is in developing this as a community resource. So it's not really, the data is not really the members of the consortium, rather it's everybody else's data. So one of the big policies we have is rapid pre-publication data release. So as soon as we have data that's been generated and it passes all the quality control standards, it is made available to the public with no restrictions whatsoever. So if a data appears there, you can download it and use it for whatever you want. In fact, there's often times where data will be made available for several years before the consortium will actually make a publication about it. So this is a really big part, and it's really great to see the community using that data. So there is, as I said, so the access to this and using it is completely unrestricted. So there really are no qualifications. All we do is ask that you reference some of the ENCODE papers about it. But you can use data that has not ever been published before in your own paper. And you can also use the software tools and analysis pipelines. Again, all of this is made open to everybody. So doing this requires a lot of the development. So for instance, we had to develop common data reporting formats, rich ontology-based metadata, and importantly, data standards. So we have very strict data standards for every experimental type of data that we generate. And if the experiment does not meet those particular standards, then it is rejected, and depending on what the nature of why it's doesn't meet those standards, the PIs then have to go back and either repeat the experiment or finish something up for that. We've developed uniform processing pipelines that everybody in the consortium agrees to. All RNA-seq data will be analyzed in exactly the same way so that you can more easily compare it to one another. And these are all run at the DCC itself. And then also analytical tools. So we also make the data available not through just posting it on the website, but through publications. So every now and then there's publications from the group. And what a lot of groups are also doing now is they've been using bioRxiv to make the publications available to the community as it goes through the process of peer review and publication. And then a lot of work is really devoted to outreach, so online tutorials and user meetings. Okay, so one of the forward-facing aspects of this is the data portal. So if you go to encodeproject.org, you'll see a page like this. In this top part here, you can actually, for instance, type in the name of your favorite gene, and it'll give you a list of all the experiments that have ever been done that characterize that gene. Or if you're interested in a particular protein, it'll give you all the assays, for instance, where ChIP-seq was done on that protein, and it'll give you a list of all the antibodies that have been tested and whether they're good or bad against that protein. And then, in addition, this is the initial interface for this screen, which is the candidate regulatory elements that I told you about, and I'll show one other thing about that in the future. There's also, this is the list of all of the data sets that are available on the ENCODE data portal. So there's over 1,500 experiments available. A bit over 9,000 of these are actually ENCODE experiments. But the ENCODE portal has also included all the epigenome read map, the modern, the mod ENCODE, the GGR, and a few community data sources as well. So it's not just ENCODE data that's available there. You can cross-reference ENCODE with other types of data. So this is the screen element that I told you about. So this is where you can search for candidate cis regulatory elements that have been predicted by ENCODE. So you can type in either a genome interval here or the name of a particular gene, and it will give you a list of all the elements within a certain distance of that it thinks are some type of regulatory element. And from there, you can click a button and visualize all of those on the Santa Cruz browser. Okay, so as I said, one of the ways that the ENCODE Consortium can make this data available to the public is through publications, and this is a slide that is famously updated all the time by Mike Pazin, one of the program directors. So this is the list of all the publications made by members of the consortium who are funded by ENCODE. But I think the most important thing is this other line. This is all of the publications, well, as many as we can find, of publications by people who are not funded by ENCODE, who are actually using that data in their discoveries. So the fact that this line has a much stronger trajectory than that of the consortium members themselves, I think says a lot to the impact of the data has had to the community in general. And also, these community publications, they span a very large spectrum of human diseases that they are actually focused on, including cancer, immunity, genetics, cardiovascular disease, et cetera. As I said, ENCODE does a lot of outreach. There's two different ways that this is done. First, there's many tutorials online where you can go and watch videos of how people can navigate the portal and what you can do with it. There's also workshops, so it's several different meetings around the US and the world. Members of the ENCODE Consortium will go and hold many workshops about the ENCODE project. And then there are ENCODE user meetings, which are occurring. This, I think, is our third one, which is occurring in July. If you go to encodeproject.org or even genome.gov. I think later today there will be an active link to allow you to start registering for one. This one will be happening in Seattle this July. And another way in which this outreach is facilitated is something Eric alluded to in his director's report, is that the ENCODE data is now hosted on the Amazon Web Services. So this means in order to use the data, you do not have to download it onto your servers locally and process it, but rather you can just open up a computing instance on the Amazon cloud and directly have access to the ENCODE data. So I think this will make a lot of analysis possible that wasn't really possible earlier. And the other form of outreach is, of course, on social media, so Facebook or Twitter. And finally, I'd just like to thank the members of the consortium. This is at one of our latest consortium meeting. It's a very large consortium of people that work very closely together and have lots and lots and lots and lots of phone calls to discuss things. And finally, I have to thank the NHGRI staff. So Elise, Stan, and Mike are the program directors who oversee everything. And Mike Pagan and Eileen Cahill are the program analysts who make sure everything continues to run. And then, of course, Carolyn, the division director. So thank you for your attention. Happy to take any questions about this.
Questions? Steve?
That was terrific. Thanks for the update. So one of the objectives that you said was to be able to have full public access to all of this. And so one is to supply the data, and you can download that freely and so on. I was just curious, for the publications, you also said you wanted to make the publications freely accessible. And is there an effort to deal with the journals to have open access and so on with those publications?
Yeah, so that's a good question. So like I said, one thing that some of the publications are doing is posting them on bioRxiv ahead of time. So that's obviously free. But I don't know if I want to use the word many or most. With one of those words, in most cases, we're making deals with the journals to make the ENCODE papers open access. I can't guarantee that 100% of them are, but we're trying to make those open access.
Yeah, I just want to say that for those of us working in this disease SNP area, it's hard to express how important ENCODE is for what we're doing. I have also been partially listening to you and also online looking at the site, and I haven't seen where you really stated that you can get access to the data on the cloud and things like that. So is that going to be advertised more broadly? Are there any issues related to data access for those who don't have accounts on Amazon or wherever?
Yeah, so it may not be easy to find it, but there is this information which does describe about how, and I think this is on the encodeproject.org website, but you probably have to find the correct
Yeah, that's where
links.
I am, so it's just hard. Like any website, there are things you can't find.
Absolutely. Yeah, I don't off the top of my head remember where it was I found it, but I'm very familiar with the website. So if you can't find it, we should work on fixing that and making it more apparent, for sure.
And it's probably my students know.
Not at all.
I know very little.
Sharon?
Thanks, and I think you alluded to it a little bit, but but I would just wanted to pick your brain a little bit more. There are a lot of people kind of outside the official funded grants or even the community, I guess, that are starting to generate more and more of this data. And so how hard is it to start incorporating really outside projects? And are there tools or are there steps of doing that to make it easier for outside investigators?
Yeah, so that's a very good question. I mean, I think the consortium would love to start including community resource or community data one of the trickiest parts about it is, like, what criteria do you say that these experiments must meet in order to be included? So, and I'm not talking about importance or anything, it's just, we have very, very clear data standards for certain experimental types. And so it gets very confusing when you have experiments that were done in a different way and processed with different standards to sort of integrate them. But I know that there are efforts that are going on to try and incorporate that type of data in a kind of a uniform way.
Right, but that can also be a carrot as much of a stick. So if your data standards are published and really clear, but it's also really clear that if you follow them, then your data can be an ENCODE or, a separate, then that encourages people to follow them.
Yeah, I agree. And I don't know if Elise or Mike want to follow up on some of this. They probably know the status of that more than I do.
Yeah, so I'll start, and maybe Mike wants to add something in. So we have brought in data from other projects like ENCODE, Modern, and RoadMap, and it's actually a fair amount of work, especially related to the metadata as well. And so it has to be, as we're figuring this out, we try to go with the ones that you kind of get the biggest bang for the buck. But I think the issues that Brent raised are really the challenging ones in terms of the data quality, the metadata, whether these are, controlled access data or not. So I just wanted to stress a point that not only is data freely available, you do not have to access it through any kind of control. There's no controlled access. This is all fully available data. So there are a lot of features. It's also a matter of bandwidth for our data coordinating center.
Right, and you could just make the submitters do more of the work, is all I'm saying, is if it's really clear what they have to do to get their data in, then they might be more willing to do it.
Yeah, the metadata is actually a very large problem. I know when they were trying to incorporate the epigenome roadmap data, even though some of the same PIs were doing it for both ENCODE and that, on similar samples, when they intersected the metadata, there were like no metadata terms that were in common. So they had to basically manually go back and redo it. So it's the metadata is a excruciatingly difficult thing to deal with.
So just one comment about being able to better deal with user-contributed metadata is that there's a number of efforts that have focused on, for one, trying to make it easy for non-experts to make good structured metadata. So Mark Musins has a project at Stanford that has developed tools for that. And then Colin Dewey at the University of Wisconsin has done a lot of work on taking unstructured metadata and automatically mapping it to standard ontologies to try and get standardization. So those are a couple of efforts you might look at there for help.
Yeah, that sounds great.
So I had a question that got emailed to me from somebody listening to this. And I was just going to say to this person, okay, I'm hoping I can represent this question. But let me just ask it anyway, because it's an Elsie question. And that is, what efforts are being made to make certain that publicly available databases are secure, especially those made available through commercial services like the Amazon cloud, given that identifiability is an ongoing problem?
To make them secure in the sense of identifiability, well, with ENCODE, that's not a problem because all of the data is open consent. So we don't use any data that has not been consented for open access release. We don't use any samples for that. Well, it's mostly cell lines, but even the human samples, like the GTEx data, there were 4 donors where that data was given with free access to make it publicly available.
And those have very limited metadata associated or phenotypic data associated with it.
Yeah.
And as part of that consent, we state methodologies change, there might be the chance for being identified.
I can't resist asking this question. Do we have any way to know or estimate how complete the catalog is?
Oh, well, that's a good question. So it's definitely not complete. I mean, I think it depends on what aspect you're talking about. So for instance, I think about half of the transcription factors have been profiled. So there's the other half that has to be completed. For the RNA binding proteins, we're only about 10% of the way through. So we have a long way to go with those. And then, I think a lot of the other types of elements have been studied much more. But again, it's really limited cell space or conditions that some of these, enhancer elements are only active in certain cell types or after a certain stimulus. So, you start to get into exponentially infinite space there. But as far as just the basic mapping of things that touch nucleic acid, I think there's still quite a bit to do there.
Yeah, thanks for this presentation. It sounds like ENCODE's been successful with the administrative and collaborative element of it with sort of uniform data sets, and it's probably been a hard-won achievement over time. Do you think that, and I think other consortia struggle with those collaborative administrative elements. Do you guys have things to teach other collaborations? Would there be value in a publication that would address the infrastructure aspect and how to set up a consortium in a more seamless fashion?
That's a good question. I do think that things work well now, and they have some things work better than others. It's been a very interesting learning experience. I started in ENCODE like 14 years ago or something, so I've been doing it long enough. It's kind of natural to me. But every time new people come in, there's a little learning curve, I would say, to get used to the way the project works. But I think once you do get used to it, you can see the advantage of the way it works. At the same time, it's kind of like a aircraft carrier. If you want to try to turn it, that takes quite a while. But some sort of publication on lessons learned, I think, would be interesting.
Mike, you want to add something?
So Jeff, this is very different than what you were asking about publications, but we are very active in trying to work with other consortia and share what we do so they understand what we do and it's transparent. And if they find that it might be of value, be it scientific, data standards, or administrative, they're aware of it and they can use that if they find that helpful, valuable.
And there are discussions with other consortia about, for instance, like the analysis pipelines. There are cases where we're trying to coordinate with them up front to make it much easier to compare data between consortia once the data generation process is done. Because if every consortia is processing things a completely different way, it makes it very challenging to do a real quality integrated analysis.
Okay, I've got Raphael and Jonathan, but are you on this topic, Jonathan?
Yeah, I just wanted to add one minor comment, which is I think ENCODE has really been great in pushing for a very rapid and free release of data accesses, as mentioned. But I think that kind of came from the ethos of the consortium. And I think that ENCODE's role in promulgating those values has been really important in the community.
So the most impressive slide you showed was, I think, for me, the one where you show the publications from others.
Mm-hmm.
Do you have a sense for what the proportion of what they're? Can you give a proportion of what they're actually using? Are they downloading the raw data, the pre-processed data? Are they just accessing a website? Are they downloading tools?
Oh, that's a good question. Mike might be in a better position. My personal feeling from looking at some of these papers is in most cases they're downloading a small number of data sets that are relevant to whatever they're actually studying, and they use it for, as sort of supporting data for a particular point in their paper.
Even for that, yeah.
It really depends a lot. Sometimes it's a browser shot. So it depends a lot on the paper. And the other thing I'd say is that line is solely from Mike Pazin collecting data, and people send to him. So if it's a very difficult job, I would say. So that's probably a large underestimate of the use of it in other publications.
So the ENCODE team works on publication tracking, and we find what we find. We're more interested in being sure that the examples we find are accurate than figuring out, did we find all or a representative set? So that's part one of difficulty of answering your question. But yeah, people use this for tool development, methods development, basic biology, and interpreting human disease. People are using raw data, people are using process data, people are using secondary websites like RegulumDB and HaploReg that have processed ENCODE data. They may not even know that they're using ENCODE data. There are things that we don't count, like if someone uses ENCODE software, we don't have a simple way to track that. So if we see that, we say that's nice, but that doesn't get added to the list. And there are non-ENCODE tools, things like, for instance, Anavar that encode and CAD that include ENCODE results in them. Since we never know, did the CAD or Anavar result rely on the ENCODE data or any of the other data in it, we don't count those either.
Yes, Hal.
Has there been any attempt to engage corporate partners that might further embellish the database, for example, with ChemSeq data?
No, not with that. I mean, I think the only real corporate stuff that I'm aware of is with this agreement with Amazon. And then a lot of the data processing pipelines are made available through, I'm spacing out on the name of that company. Oh, DNA Nexus. Yeah. So those are the two main corporate partnerships that I'm aware of.
Okay, Brent, thank you very much. Wonderful presentation. We're ready for lunch, but first, council photo is due. So if you would just gather, if you would just gather in the atrium of this building. I think there's a place that's been selected for the. We're going to do that right now. We're going to do that right now and then come back for your lunch.